Narrator. In the cutting-edge world of cancer research, a fascinating player is changing the game: extrachromosomal DNA, or ecDNA. This tiny circular DNA found outside chromosomes is opening new doors to understanding and treating cancer. Let's dive into how xDNA is revolutionizing oncology. First, what exactly is ecDNA? Unlike chromosomal DNA, which follows stable inheritance patterns, ecDNA is distributed randomly in cancer cells. This randomness leads to high variability in gene copies, often containing oncogenes and regulatory elements like promoters and enhancers. In simpler terms, ecDNA can dramatically amplify cancer-causing genes, fueling tumor growth and speeding up cancer evolution. Now, how does ecDNA shape cancer progression? The presence of ecDNA is far from random. It varies across cancer types and is often tied to cancer aggressiveness. For instance, ecDNA appears in over 50% of liposarcoma cases and nearly half of glioblastoma and HER2-positive breast cancers. The amount of ecDNA can even fluctuate under treatment pressures, allowing cancer cells to adapt, survive, and resist therapies. Studies like the 100,000 Genomes Project reveal that ecDNA is detectable in about 17.1% of tumor samples, correlating with tissue type, genetic mutations, and cancer subtypes. This irregular distribution gives tumors a powerful evolutionary advantage, promoting rapid changes in gene expression that help cancer cells evade treatments and adapt to their environment. One of the most intriguing aspects of ecDNA is its role in therapy resistance. Unlike chromosomal genes, ecDNA can independently increase or decrease gene copies, enabling tumors to produce more of certain proteins that promote survival under chemotherapy, targeted therapy, or immunotherapy. For example, after treatment with temozolomide, TMZ, glioblastomas often show an increase in ecDNA carrying EGFR mutations, which enhances the tumor's adaptability and aggressiveness. Moreover, ecDNA often includes genes involved in immune regulation, suppressing the immune response. Tumors with ecDNA are more likely to evade immune surveillance, creating an environment less conducive to T-cell activity, potentially reducing the efficacy of immunotherapies like immune checkpoint inhibitors. Essentially, the more ecDNA a tumor contains, the less likely it is to respond well to these immune-boosting treatments. Given its crucial role in cancer survival and evolution, ecDNA is gaining attention as a promising therapeutic target. Researchers are investigating ways to specifically recognize and degrade ecDNA, or disrupt its formation and distribution. For example, drugs that block ecDNA replication could prevent cancer cells from maintaining high copies of oncogenes, potentially slowing tumor growth. Enhancing DNA repair processes within cells might limit the formation of ecDNA, reducing the evolutionary flexibility that allows tumors to become more aggressive or resistant to treatment. Monitoring ecDNA levels and changes could also help tailor treatments more precisely, paving the way for personalized cancer therapies. Understanding ecDNA's function and formation is essential to advancing cancer treatment. By targeting ecDNA directly, researchers hope to reduce tumor heterogeneity, slow disease progression, and overcome treatment resistance. In the coming years, ecDNA research promises to transform cancer therapy, making personalized medicine more effective in improving patient outcomes.